Thanks, Dr. Tanaka introduced, and thanks this opportunity to present our data here. But I should say a little sorry about it. Since this title looked like they want to analyze this genome-wide like a phylogeny analysis, but actually we want to show that this idea here today. So as a before yesterday in the morning, there are some professor mentioned that how genomic search to contribute to plant breeding. So I added this slide yesterday. And so here is the situation that we have to concat this idea of the RepDB to make a foundation of the genomic research that to annotate genes. We like to make a basic data or marker database to contribute our plant breeding program. Here I take some example that show here is some variety in Taiwan. In the upside is blue color means japonica, and the red side and below red color means indica. And sometimes they want to maybe they set up some breeding program, and they make the cross between the indica and the japonica, but also sometimes the indica or japonica. And in this case, in some cases, they need more marker to analyze it. Since 2005, our government big support to our marker assistant selection. So start from that time, many research government research institute have the power to do the marker assistant selection. So they need more molecular marker. So based on the published data, they can find that we have the Nippon sequence and another indica sequence code is from the variety called 9311. But sometimes it's not enough, and so we contribute our other sequence data to make more condensed marker data to them. And also another advantage is our variety all come from the Taiwan. Come from the Taiwan variety that we discuss and chosen discuss with our plant breeder in different breeding institutes, and also we check that based on their pedigree. So when we check the pedigree of our variety, we found that sometimes our japonica rice they have some indica background. For example, here I take this Tiger 9 as an example. The Tiger 9 have the high eating quality variety in Taiwan, and the most convenience store like to use this to make the lunchbox. And in his pedigree, we found that its male parent is indica. And in this pedigree, we can find that the famous TCN1, that's Taichung native 1, the variety with the semi-dwarf gene. That's with from 地角物间. I think you may know. So in here we found that based on this pedigree, maybe you can calculate the coefficient of the relationship between the indica and japonica, that's 0.5 and 0.5. Actually not, because they have sometimes they will make some recurrent selection. So, but now we can see the pedigree. No, sorry, we can see the coefficient of the relationship based on its pedigree. Here, the bigger number means the relationship more close. So you can see here the Genome 67 that we made the train population variety. This very close to Taichung 65. And with the Tiger 9, you can see that this coefficient is very low because half of it is indica. And later we can find the result of the SMP analysis. It can all twist this result. So as I just said, we discussed with our breeder, and based on the pedigree, we chose the 7 Taiwan modern variety in 5 japonica and 2 indica. This in here, and we use the sequence technology GA275 pair N in beginning for the 7 variety, and later because the Genome 67 is very important to our research, genomic research in Taiwan. So we do additional sequence in 101 pair N for one length. And our reference genome still use the IRGSP5, and I will change to the Genome 1 quickly. So here you can see that after we got the sequence data, we do some preliminary analysis here. So the total base, like here to show, that's 2.7 to 2.9 gigabyte. Is that the Genome 67 because of the 101 pair N. So we up to 13.2 giga base. And we map to the IRGSP5 as the reference genome. We can see that the mapped rates from 89% to 96%. And here you can see in this red color means indica, and you can see that even in the almost the same quality as other japonica, you can see that they are less 5% you cannot map. And this part maybe I think that maybe just the difference between the indica and japonica, and we will do the analysis later. Hope I can show that data for you later. And here you can see that as we know as from the pedigree, the SMP number in 1 mega, the Taichung 65, they are 241 compared to Nippon berry. And India we got so it means it's very close to, because it's a minima here too. So I think this in this you can see that it's very close to the Nippon berry. And here you can see that the Taichung Xian 17 it's have 4,855. So this means that's more far away from compared to other variety. And here very strange thing that maybe you notice that 1067 got a 89 Intel uh, compared to the Nippon berry. That's I think this part maybe because of the coverage, more coverage, and because our Intel filtering is the criteria is high, is somehow maybe too high, so the data show here. So I don't take this as the distance to Nippon berry. And after we got this basic status, we put in make a program to calculate the SMP count every 2 million base compared to based on the Nippon berry, the reference genome, and we make this spectrum here. Here you can see the color red means the Taichung 65. Taichung 65 in this chromosome 1 somehow is very low here. And Tiger 9 in the beginning are very high here. And this light blue and the purple color means 2 indica. And they're always in the top. Sometimes, somewhere for this japonica rice, Genome 72 in this area are very similar to indica, and have a very high SMP count. And here also a little small part, this 3 japonica are more close to, more far away from the Nippon berry. And here we compare to the results is published by this group. I think this Tokyo University and the NGS group. They published in 2011. They show that they use some land race in Japan and do almost the same thing to analyze the SMP count or the frequency. Show in red part, they show that very high dense SMP here, and the blue part very low SMP part here. And sometimes it's very our results are in the this 7 Taiwan variety are very close to their result, but sometimes are not. And in chromosome 12, the situation is the same. Sometimes our japonica rice have this shape. It's look, so in this area, it's look like very close to indica, and in this area, it's not. And compared to the result, and compared to the Japanese result, we have some difference in here. For example, this Genome 72 have a high SMP dense here, but low in here. And the red color means the high dense in Japanese group result. So compared to them, we almost the same. Compared our result to their result, I think because they use the 1 mega window and non-sliding, so somehow our I used the 2 mega window and sliding about 5 kilobase. So sometimes our result will respond very slow. So maybe later I will change the different window size to check if we are same or not. And before that, we use the all sequence compared to the Nippon berry. Here I show you that we compare to 1-to-1 for 7 variety. Oh, of course, compared to including the Nippon berry. So 8 variety. Here I count that the distance between two SMP or indel. So the bigger number means they are more close. And so, but you can see that very logically that it's reasonable that the japonica have a high number and the indica have a low number here. And compared to the pedigree, based on the coefficient of the relationship that based on the pedigree, you can see that somehow, take the Tiger 9 as an example, you can see that it's not very far from the, actually it's not very far from the Nippon berry here. And we use this SMP result to draw a phylogeny tree here. And so you can see that it's a very clearly these two indica are very far from our japonica. And if you see here, you can see that the two indica are very close to each other, but actually when they compare to each other, it's not. Their distance still very far to each other. And as I say that we want to contribute our genomic result to our plant breeding researcher. So we construct the data about the indel size about 15. That's easier to our research. They can use to design the primer and analyze the difference. So the number we show here, the distribution like this. And later I will provide this table to our research like this, and we have this also provide the sequence in this indel side in the right and left side for 300 base pair. And we check, before we deliver this data to our research, we also check in IGV. You can see here, for example, Taichung 65 compared to 2 indica rice. Here you can see that there is no indel here, but in these 2 indica we found these 2 deletion here. And also we check the research assistant in our lab to do the gel analysis that they got we have confidence that we have almost about 80% accuracy. And we found that one young breeder to help us to check our results. They use 2 different indica and japonica, and their hybrid program that's almost about 66 accuracy here. And sorry, I take too long time. So that's the slide. I want to thank our lab and several people here and our breeder to contribute our rice breeding program.
Thank you, Mr. Wei. Because of the time schedules, I move to the next speaker.